Hello, I am Frank Helle and I'm a biology teacher at Bergen Katedralskole in Norway. One thing my students struggle with in biology is data handling, especially statistics. In this video, I'm going to show you on a practical level which tests to use in your essays. But mastering this is important, however, since statistics comes in under the analysis criterion, which is 6 out of a total of 24 marks. In the extended essay, statistics is within the analysis heading under this C criterion, which is an astounding 12 marks. But not only that, having good and well thought statistics can even affect the marks on other criterions on your study. But why does IB have this emphasis on statistics? Well, let's say you just discovered polyhedral dice and you want to check out the D6. You don't have the math yet to know that its average is 3.5. So you do some measurements. You get a 3, a 4, a 5, divide by 3 and you get a 4. Then you see fascinating D12. You measure it as well, you get a 1, a 4, an 8, divide by 3 and get another 4. So you conclude that the D6 is as good in giving high numbers as the D12. But in reality, the average of the D6 is 3.5 and of the D12, the average is 6.5. So you've made a wrong conclusion. And this is what statistics in biology is all about, how to avoid making the wrong conclusions. So let's start with the red die. So the first one was a 1, then the average is 1. Then you go to the second row and actually now your average is correct. The third you go a little bit over, and as you continue making the fourth, fifth and many more rolls, you see I have an exponential scale on the x-axis. You will over time get closer and closer to the true mean, and you will end up after 100 rolls with a mean about 3.5. The same will happen with the D12, but you had a pretty poor start. So the average will probably increase more and more, and as you get close to 100 rolls, the mean of the D12 will be close to 6.5, meaning that if you do many repetitions, your measurement will always be closer to the true mean. It turns out when you have two dice, some combinations are more common than others. We see that the value 7, for example 4 plus 3, have 6 different combinations, while the result 2 only have one combination. In other words, the values from rolling 2 dice will have something that approaches a normal distribution. And this is a very important principle in biology. And the same is true when, for example, measuring the height of a plant. There are so many different factors affecting how tall a plant gets, such as light, wind, carbon dioxide excess, pathogens, toxic minerals in the earth, and so on. And each of these variables can be regarded as a die. And the most frequent measurements when you measure the height of a population of plant will be kind of the 3 and 4 values of a D6. So I have actually some pea plants in my garden. This is 1 out of 4. But let's say I have, instead of 4, I have 50. If I measure them, it would probably look something like this, with an average in the middle. So each of these dots represents a height measurement. Adding them all together divided by 50 would give me the L bar for the length outer plants. And those values would probably follow a normal distribution. But I would never have the patience to measure 50 plants. I would maybe only measure 8. And that would be a sample of the whole population. And by chance, I could maybe take some low plants and get a sample mean that is lower than the true mean of all the plants. Or I could maybe by chance pick some tall plants and get a higher sample mean. Let's say I also have 50 plants in greenhouses. Then, if I took all the plants in the outside group and pair them randomly with all the plants in the greenhouse group and calculated the difference, I would get a new population of values which represents the difference between greenhouse and outside plants. If data are unpaired or independent, it doesn't matter which values we match from the outside and greenhouse plants. If data are correlated or paired, however, it's very important that we subtract values from the same plant. The reason for this is that this will lower variation, because if a plant is tall to begin with, the delta value, the difference, will be smaller than, for example, if we matched a tall and a short plant. If the means of the outside and greenhouse plants are close, that would make the normal distribution of the delta values closer to 0. In contrast, if the distance between greenhouse and outdoor heights are high, then the delta values would be far away from 0. But this population would have one important characteristic called standard deviation. So let's say I have 8 measurements of each plant, and let's say I took the distance from each measurement to the average, L bar delta, and then squared that value so that instead of a line, I got a square. Then I added all these squares together, divided by the number of measurements, 8, and took the square root of everything. That will be the standard deviation. Showing it with mathematical notation would look something like this. And it turns out that if you have exactly 1.96, so almost 2 standard deviations going out from each side of the mean, you cover 95% of all values in the normal distribution. Meaning, if you just pick the number of plants from the population, it's a 95% probability that the average would be in the red area here in this graph. And only 2.5% chance that the average would be on one of the tails. And this is important because we use this confidence interval to distinguish between 2 hypotheses we always ask in biology. The null hypothesis, or H0, is that plants growing outside have the same height as greenhouse plants. The H1 hypothesis is that plants growing outside have lower height compared to greenhouse plants. And so here again we have the outside and greenhouse plants, and then you have the delta values. So if the 0, here shown with a black marker, is outside the confidence interval, we reject the null hypothesis, we adapt the H1 that the outside and greenhouse plants have different height. If the 0 is inside, however, we keep the null hypothesis and say there is no difference. Actually, according to height, these plants belong to the same population. Let's look at 3 different situations, A, B, and C. In situation A, the normal distribution of the values is so that only 1% of the graph, or the area of the graph, is to the left of the 0 and 99% is to the right. Then we can calculate a characteristic called the p-value. To calculate the p-value, we take all the possible averages where 0 is included in the interval and divide it by the total 100%, and we get 0.01. And what this means is that if we did this experiment 100 times, 99 out of these times it would show the height of the outside plants smaller than the greenhouse plants. Another situation would be that we had 5% of the curve, which would give a p-value of 0.05, meaning that out of 100 experiments, 95 would show lower mean height of the outside compared to the greenhouse plants. And finally, in situation C, we have 15% to the left of the 0, which means a p-value of 0.15. But which of these are statistically significant? Does data need to be so good that we only see kind of an erroneous result 1 out of 100 times, or something else? Well, the answer is A and B are significant, but C is not. Why is that? It's because in biology we have adapted an alpha of 0.05, meaning that as long as the p-value is 0.05 or smaller, we say that the result is statistically significant. In other words, p less than 0.05, it means we accept the H1, the alternative hypothesis, that the populations have different height. But if p is larger, we keep the null hypothesis that these two plants actually belong to the same population, at least in terms of height. You might wonder, in example B, there was a 5% chance that we could actually measure the average in the greenhouse plants to be lower than the outside plants. How is this possible? Well, it can happen by chance. Let's say I pick only tall plants from the outside population. We see the red crosses here. So we see they are picked from the right tail of the normal distribution. But it could also happen at the same time that we picked the greenhouse plants from the left tail, meaning the lowest plants from the greenhouse. There are other tricky things we have to consider when doing statistics. One problem, which is shown here, is that the standard deviation can vary between populations. So for example here, we have small plants, but they vary in size. Let's say this plant might be this one, this plant might be this one, for example. The other populations have taller plants, but they vary less in size. And differences in standard deviation can kind of confuse many statistical tests and give wrong results. Another problem is the data are skewed, that you have more values close to the left tail than the right. Compounding these two errors, normally not a problem in itself, is that you have different number of values in each group. For example, here you did experiment with boys and girls, and more boys wanted to partake in the experiment than girls, so you have this difference in numbers. And if you have combinations of these two or even all three, you are often in trouble, and the examinators might spot it, so you better comment on it. The type of data you use in your IA or EE determines the statistics. The most common type of data is numeric data, like we have just used in our examples. Like, for example, length, for example, in centimeters. But these data, numeric data, can be paired. That is, if the subject is the same but have two different treatments. Or data can be unpaired, like we did with the pea plants, because there different plants grew in the greenhouse or outside. The other type of data is categorical. For example, disease or no plant disease. One type of categorical data is ordinal. For example, the growth stages of pea plants, which can be non-deterministic, like number 3 here, or just going for the light as fast as possible, which is number 1. And you can order them after how extensive they are. But you have also just nominal data, for example, colors, data that hardly can be placed in order of each other. And the subcategory of this is binomial data. And after this video, I hope you will understand this table. But to be frank, just doing a paired t-test could be enough to get you the best grade in either EE or IA. But if you had a little bit more complicated design, like you had 3 groups here and did an ANOVA, it could sometimes score you another point. But the most important value of this video is helping you avoid the pitfalls by pointing you to the correct test. If you, for example, have skew data, you might have to do a Wilcoxon signed rank test depending on your experiment. or maybe you have to do a Fisher's exact test. Just knowing the statistical test to choose isn't enough. When you design your experiment, you have to do it with the test in mind, because it is possible in many ways to design experiments that you can't test statistically. Then, when you perform the test, you have to look for trouble along the way. For example, skew distribution or uneven standard deviations. And if you see these problems, try to take action or be prepared to use more exotic statistics if you don't. Then you have to document very well what you have done. So you have to include a statistics paragraph in your results or analysis paragraphs. Here I have shown an example for the famous potato experiment in 2IB. Also, you have to mark the outcome of the statistical test. For example, like I've done here in the main process data table. And then, most importantly, when you conclude, you have to base your conclusion on the outcome of the statistical test in addition to the numerical difference in averages. And so we arrive at the charts of the statistical tests and characterizations. I think will cover most of what you need when writing your essay in the IA or EE. The goal of the test is here in this column to the left, while the type of data are in horizontal columns along this chart. We start with number 1, which is how to characterize a population using the mean. But to do that, we first need a mean. So we will take pea plants that have grown with bokashi fertilizer, and then we click on how to get basic stats from a web page by Richard Lowry, which is called VassarStats, which we are going to use a lot. We paste the values, take care not to have an empty row, press calculate, and we will get the number of measurements, the mean, and the standard deviation, as we talked about, and something called the standard error, which is more a description of where you can expect the mean of 8 measurements. Number 2 is if you want to compare a mean to a specific value. In my case, I want to see if the bokashi plants have the height which is promised on the packet of seeds. So I start the test and paste my bokashi data one more time, and I write the value on the package which is guaranteed that the plants would be 15 centimeters. I press calculate, and I find that the sample mean 13.25 is indeed below the hypothetical value or the promised value of 15. And here we have to look at the two-tailed test. Remember, one-tailed is only used if we have very good reasons for it, but the two-tailed p-value says that almost always, if I measure my plants, they will indeed have a lower average than 15. So I can complain to the seed producer. The second test is a two-sample t-test. I select the data or heights for the first group, the bokashi plants, Ctrl C for copy. I go to the t-test, and since these are independent samples, meaning it was different plants living under different fertilizer conditions, I press independent samples. If instead I measured growth per day, for example during sunlight or overcast, but the plant was the same from day to day, then I would have chosen correlated samples. But here it is independent samples. I paste the bokashi data, take care not to have an empty row, go back, select the bone meal data, paste, and then press calculate. And we see that the average of A, which was bokashi, is 13.25, but the bone meal plants were higher. But was the difference significant? Yes, if we look at the results column, we see that the p-value, two-tailed, and remember always choose two-tailed, was 0.001, which is highly significant. This test, ADF test, just checks if the variance, the standard deviation, is different, and luckily it wasn't. This t-test would have been the one we would have to use if the variance was indeed different, but it would still have been significant. In the next test, I want to include pea plants that have grown with compost fertilizer. And to do this, I cannot use a t-test because it can only compare two groups. I have to use an ANOVA. So I start the test, and I have to say that I'm going to compare 3 samples. Since they are individual plants, they are independent samples. And I also have to say that it shall perform a weighted analysis. This is a good choice because it's more conservative, and so you always choose weighted if you are in doubt. And then I see that I have to copy the data column by column. So I start with 1 as the bokashi group, press calculate, and here we get the number of data in each population, the mean, and we see that the mean of the compost is 19, higher than the 2 others. We get standard deviations and standard error, like we are now used to. But are these data significantly different? Yes, they are. The p-value for the whole set is 0.001. And this just means that the population with the highest and lowest means are significantly different. So in this case, sample 3 was compost. It is significantly different from the bokashi group. But what about the bone meal plants? To know if they also are statistically different from the other groups, you have to look at something called a post hoc test. In this case, it's called a 2K. And what it does is that it checks the groups with averages in between the extreme values, or the maximum and minimum. And here it tests M1 bokashi versus M2 bone meal. And that comparison is indeed significant, meaning that there is a statistical difference between bokashi and bone meal. Then it checks bokashi and compost, and that was what was done here, so we already knew that. But then, more interesting, it checks M2, which was bone meal, versus compost, and that was also significantly different. So in other words, all the fertilizers had a significant effect on plant height compared to the other groups. But it turns out that I have good room in my greenhouse, and I've tried the 3 types of fertilizer, both inside my greenhouse and outside. So now I've kind of 2 nominal factors, inside or outside greenhouse, and type of fertilizer. Those 2 are nominal data. And then I have plant height as a numerical variable. So we can see that this is kind of a 2 by 3 factorial design. And to do that comparison, you have to use something called a two-way ANOVA. So number of rows are 2, which means inside or outside greenhouse. And number of columns would be 3, the 3 fertilizers. And now I have to press setup. And then we, as always, we have to choose weighted. And so we see that there are 6 columns here to place the data in. So sit back while I copy the data using 4x speed. And then these values comes in the second row, since they are in the greenhouse. And now I have to check that there are no empty rows. in these fields. It looks like I'm good to go, so I press calculate and hope for the best. And indeed, I got data for all the fields. So as usual, we see the Bokashi outside. All of these are known averages. And then we see that inside the greenhouse, all values seems to be higher. So the Bokashi greenhouse pea plants are 20 centimeters, much taller than the outside plants. And we see that this is true for the bone meal and compost plants as well. But are these data significantly different? And we compare two things when you have factorial ANOVA or a two-way ANOVA. One thing is that you see the effect on the rows. The other thing is that you see the effect on the columns. And if we look at the row, it says that the HSD, or the absolute difference between the two means, should be almost 1 for a p-value of 0.05, or 1.32 for a p-value of 0.01, but since our alpha is 0.05, we look at this value. And if we look at the row difference, we see that it's 15 on the outside plants and 22 on the greenhouse plants. So we see that the difference is much greater than 1. So there is indeed a significant effect of greenhouse, no greenhouse. But now we want to look if there is a significant effect of the fertilizers. And that means that the difference between the columns should be 1.46. And when we look at the Bokashi mean, it's 16, while it's 18 in the bone meal group. And this difference is 16 to 17 to 8. It's 1.8 approximately. And that's more than this minimum value of 1.46. So there was a significant difference here. And then if we look between the bone meal and the compost, there is also a difference greater than 1.4. And this is confirmed when we look in the ANOVA summary table, which says that with a p-value of 0.0001, there is indeed a significant effect between the rows, meaning outside inside greenhouse, and effect of fertilizer. If you have uneven standard deviations, as I showed in a previous slide, meaning that the standard deviation of one group is very different from another, then you have to use Welch's t-test. Here you come to a graph pad tool, you choose enter up to 50 rows, and you choose Welch unpaired t-test, and then you can label the two groups. In my case, it's Bokashi and bone meal. And then I add each single value here. And when you have entered the values, you press calculate now. And we get the p-value, which is 0.0025. If you are doing ANOVA with greatly different standard deviation, you can use Welch's ANOVA. To do this, you have to open a spreadsheet made by Richard Lowry, and we see that we have some sample data here. So we exchange these CTs with fertilizer, and then whatever it was with plant heights, we get the recognizable means here, and we get a very small p-values indicating again that these values are significantly different. In the past examples we've had one nominal category such as fertilizer, and then a numerical one like plant height. But you can also have two numerical variables, and to compare them we usually do a regression. And one of the simplest is linear regression. So we click number 8 here, and we come back to VassarStats. And the thing we do here is that we paste the x and y variables of the regression. So in this case, we copy water amount that the plants get with their growth speed in millimeter per day. We paste the data directly into the field here. Take care not to have any empty rows. And we get all these delta values. But the important thing here is firstly the r squared value. And this value tells us how much does the one variable, for example water, depend on the other, which is growth. If this value is close to 1, it means it's a very close dependency. If it's close to 0, there is almost no dependency. This value 0.95 means that in this situation, if you have one value, for example the water level, you can with great certainty predict the other value and vice versa. But is this r squared value, which is significant since it's 0.001 less than 0.05? But what if you want to compare to regression? Let's say we do this for 2 plants, pea plants and tomatoes. And then we want to know if these 2 regressions are statistically different. To do this, we need an ANCOVA. And we choose the data import version. And since it's 2 regressions, we choose k equals 2. And we first paste the data for the pea plants, which are similar to number 8. Then we get the data for the tomatoes. Remove the empty row, of course. Press calculate. And we see that the means are very different. 23 for the pea plants and 41 for the tomatoes. So this first box here, ANCOVA summary, tells us if the intercepts of the 2 different regressions are different, and they are. If we look at the second box, which says homogeneity of regressions between regressions, we see that the p-value is not significant, since it's bigger than the alpha of 0.05. What does this mean actually? This second one means that the slopes of the regressions are not significantly different. So the slopes are kind of parallel. But this one means that the slopes crosses the axis on different values. If we couple that with the averages of 23 and 41, we probably have 2 parallelish lines where the tomato one is much higher up than the pea plant one. And they are indeed different. So the relationship between watering and growth in the greenhouses do differ from tomatoes and pea plants. The third thing we can do with regressions is to look at 2 independent parameters, how they affect a single dependent parameters. Independent is the thing that you change in an experiment, and the dependent is what you measure. Usually in IB you just change one thing, but in very special circumstances you might change 2 things and look at the effect. And this is what you use a multiple linear regression for. So let's say in addition to watering levels, you want to look at the effect of sunlight. And so you have to put all the rows in here. And how do you arrange them? You put the x1, independent one, then the x2, and then the resulting dependent. So we take the and move the growth speed, the y variable one to the right, so that we can fit the x2 in beside. So this is x1 water, what we measure, and this is x2, and then this is y, the effect. So here we have the data. We paste, remove the empty row, press calculate. We see that the regression weight of x1 is very high. That is the water, while the regression weight of the sun hours are quite poor. But we see that the regression as a whole is still significant, but maybe it wasn't very useful to add the sun data. The fourth type of regression we can do is logarithmic regression, and that is when there is not a linear relationship between the two variables, but a logarithmic one. And first we have to scroll past a very good example of how to use logarithmic regressions. But here we come to the data entry. So here in the X you can have the logarithmic input, and then instead of numerical values on the Y, we have to put in outcomes 0 or 1. In my example, we are using bone meal to combat a mildew infection. So the bone meal provides nutrients that strengthens the immune system of the plant. And so here is a bone meal in a milligram per kilo soil. And we see that with the lowest dose, only 2 are healthy, while 8 have mildew and so on. So now we take this data and transfer it to the page. And when we are finished, we press calculate. We see from the r² value that the regression follows very close to data. And we get a very significant p-value indicating that the protection conferred to the plants by the fertilizer is dose dependent, meaning the higher the bone meal dose, the better the plant is protected against the mildew infection. The last test on numerical data we are going to look at is the outlier test. Let's say you have many values close to the average of the sample, but then you get suddenly a measurement which is way off. A good way to exclude it from your data as an outlier and probably get marks in your data handling skill is to do a Grubbs outlier test. The way you do this is that you start the test and choose an alpha of 0.05. We take all these data, paste them, remove the empty row, press calculate, and see if we find an outlier. And so here each value is evaluated, and indeed row number 9 contains a value indicated by the Grubbs test as an outlier. Until now, we have looked at situations where one variable is categorical and the other numerical, like the t-test, or both are numerical, like linear regression. Now, first, we are going to look at a population where both variables are categorical. To describe one population of categorical data, we use the median and the interquartile range. We use this page from Math Portal, and we get the data from our spreadsheet. So these are the growth stages of a population of pea plant growing with bokashi fertilizer. We copy, paste, and first we can choose median to see that the median is 1, or we can choose the interquartile range, which is the third quartile minus the first. This can be easily done by hand as well. The next thing we're going to do is compare two populations of categorical data. And the first thing you have to do is input how many data you have, and in this case we have 12 pea plants in each group. And so we have to paste sample A, bokashi plants here. Again, we use the bokashi growth stages. Then we look at the bone meal growth stages. And then we have to press import data to data cells. So here these have been sorted, and now we press calculated from raw data. And what this webpage does, it combines both bokashi and bone meal plants and mark them if they are from A or B, and then they sort them chronologically, and then they are given a count from 1 to 24, since there are 12 times 2 plants in total. And if they share a rank, for example, we see that there are 1, 2, 3, 4, 5 plants that have 3. If they share a rank, they are given a shared rank, which is the average of rank 24 to 20, which is 22. And so it goes. We see that the sum of ranks for bone meal is 105.5. And what we are going to look at is this value different from the population consisting of both sample A and B. So kind of this test is special in that you don't test if A is different from B, you test if A is different from the AB combined population. If this value is higher than the upper critical value or lower than the lower critical, the result is significant. And you find these lower and upper limits here in this table. And if we look at the directional test, it seems like our results is statistically significant since our UA is higher than 102, meaning that the p-value is 0.05. But we cannot use a directional test. Remember, I couldn't know beforehand that bone meal would always be big, have higher or more advanced growth stages than bokashi. I couldn't know that beforehand. And that's what you do when you do a directional test. You say, bone meal must have greater growth stages than bokashi. Sometimes you can do that, but in this case, I can't. So I of course have to do a non-directional test. And I see that my UA, it falls within the 0.05 range. So it's lower than the upper limit of 107. If my sum of ranks had been 108, for example, then my result would have been statistically significant. If you are comparing two populations of categorical data that are correlated, however, you should use a Wilcoxon test. First, we go and look at the data. So this is my garden in 21 this year, but last year I got a lot of family and friends to rate their engagement with the garden in July and then in August. And my hypothesis was that people were more engaged with the garden in August because then they could get produce from it. Like many of my friends, they collected salads and such after being on a visit. So we copy these data and then we choose the Wilcoxon test and we can paste both columns in here. Mind the empty row. We have to first import the data to these cells and then press calculate. And we have to look at the two tails as always, and we see that there is indeed a significant increase in rating from June to August. We see that the W is minus 97, and that is actually an increase because if we look at this column here, it's the June values, or was it July, minus the August engagement ratings. So this sum being negative means that the August numbers or ratings were higher. To compare more than two groups of categorical data, you need a Kruskal-Wallis. We first go to Excel to look at the data. So we see here we have a third population of achieved growth stages, but these plants have had a compost fertilizer, and there was 12 bokashi plants, 12 bone meal, and 12 compost. and we need to copy the columns one by one. We press import to data cells and then calculate from raw data. And we see that, similar to the Mann-Whitney, all the data are marked from which sample they belong to, but then put together and given a rank. We see that the p-value is 0.04. So we see that there is a statistical difference between the groups. If data are correlated, however, you use a Friedman test. Here k equals 3, but I think there are also other options on the VassarStats page. K means how many samples you compare. And we expand on the engagement rating, and we ask the participants how engaged they are with Frank's Garden during December, and we see that there is 17 measurements. And in order to paste whole columns of data, we have to activate this JavaScript, and now we can take one column, can import column by column. We press import to data cells. So here we have the raw data. Now we can go with calculate. We get a low p-value indicating a significant difference between the months. The next type of data we are going to look at is binomial. And if you have a population with binomial data, you can just give it as the proportion. For example, 3 out of 5 plants were green, for example. If you're going to compare population of binomial data with an expected fixed value, let's say you are counting yellow and green plants in the F2 population, and your Punnett square says that it should be 50% yellow, 50% green. We can look at these data here. So we got 58 green plants in the F2 and 42 yellow. And so to see if this is within expectation, we go on binomial data. And it was in this experiment 100 plants, and we saw 58 of the green variety, but the assumed frequency was 0.5, and we get a p-value of 0.13. This is larger than the alpha of 0.05. So this means that even though we saw a suspiciously high number of green plants, 8 more than expected, these values are within the range of this experiment. So the null hypothesis concerning the Mendelian genetics of the Punnett square still stands. If we are going to compare two different groups, however, we need to use a Fisher's test. And in this case, we are going to look at mildew data. If I have a mildew outbreak, and I would see if fertilizer had an effect on number of infected plants, I would go on Fisher's test. And we see that here we have, for example, fertilizer on the X, and then mildew infection status on the Y. Let me show you the data. So here is a healthy plant, and here is a mildew infected pea plant. And in the Bokashi group, I mark all the infected plants with a 1 and the healthy with a 0. So we have 12 infected in the Bokashi group. So there are 22 in total, so that means 10 are healthy and 12 infected in the Bokashi group. But plants with bone meal fertilizer, there's 17 are healthy and only 5 infected. So 10, 12, 17, 5. Let's see if we can remember that. Bone meal healthy were 17 and bone meal infected were 5. And now we press calculate, and this table shows the expected frequencies based on total number of plants and total number of infected. And so what this test does is that it substitutes the expected frequency from the actual frequency. And we get a p-value of 0.06. So the effect of bone meal was non-significant in this experiment, meaning that the differences between infected and non-infected are not great enough to exclude a random variation in data. But sometimes you have more than 2 categories. Then you don't have binomial data anymore, you have nominal data. And as long as the outcomes is always above 5, so you have no value in the factorial design which is less than 5, you can do the chi-square test. And the number of rows was fertilizers, bokashi, bone meal or compost. And the number of columns again is infections, infected or not infected. So we see here the cells, and now we will look at the data. So we have continued on the mildew example, and we have added the compost fertilizer, in which 14 plants were infected, and 8 and 14. We go calculate, and we see that the p-value is indeed significant. And this probably has to do with the bone meal group, which is deviates positively in terms of healthy plants from the total. We can see also that compost for some reason is really bad. So maybe the mildew infection is in the dead plant material of the compost. The next thing we are going to look at is survival analysis. Let's say you have some kind of hazard that kills stuff. In this case, I have UV, outside UV light on my pea plants. And to analyze this, we use a Kaplan-Meier. But first we have to look at the data. So I started off with 50 pea plants in the garden. And then I have sparrows that are looking for worms, and then if they see a small plant, they rip it out of the soil. So when I saw this happen, I censored the data because these plants were ripped out by sparrows, not died from UV exposure. And then here in this column I have the plants that were indeed dead by UV. And so we see that here 6 are missing, so we are left with 44, and then 3 died, and so on. So we also see that there is 1, 2, 3, 4, 5, 6 periods. We start the Kaplan-Meier, and the first thing we have to answer is the time periods, which were 6. And then we start with 50, and then first we lost 4 to sparrows, then 2 died, 0 and 3 and 10 and 5, 5, 3. And here we get the survival probability estimate. What this means is that, for example, if to wait 4 time periods, so let's say this is 4 weeks, then the survival probability is 0.51 or 51%, and so on. 6 days is a survival of 25%. So there you have it. Now we have been through 22 working and 1 non-working statistical test. And this chart here will be available as a link in the description. So Whenever you want to test your data for your extended essay or internal assessment, go and click here and you will arrive at the appropriate test. Maybe watch this video how to perform it. And then, in addition, I want to show you the Handbook of Biological Statistics. This is a very good resource when you're going to write the statistics chapter, where you explain the rationale and choices for your statistical test. It's very well written and very much to the point, like very practically oriented. So write in biostathandbook.com or just follow the link in the description. With that, I wish you good luck in all your endeavors writing the big essays of 2 and 3 IB.